let's talk about primary homeostasis. So primary homeostasis ends with the formation of platelet plug, and it is a really weak platelet plug that is formed, and it is then stabilized by coagulation factor. So the end result of primary homeostasis is the formation of platelet plug. How is it formed? We will talk about next. So whenever there is a trauma to the vessel, so for example, here we have the endothelium, and here we have the endothelium, and surrounding the endothelium we have smooth muscles like this, around the vessel. And then, so the first thing that occur after trauma to the vessel is vasoconstriction, transient vasoconstriction. And the vasoconstriction is mediated by, number one, by endothelium. It kind of causes the smooth muscle to constrict, and then cause the vessel to occlude the lumen, and so that kind of results in decreased bleeding. So this is the whole idea behind transverse vasoconstriction, is to prevent bleeding. So for example, if we have there's a trauma here, and the remaining vessel is this, and this whole thing is removed by trauma, then blood will be coming out of here, these areas. And in order to stop, blood is coming through this way and then will be coming out. In order to stop that blood from going out through the traumatized area, is to cause vasoconstriction, and this is transient. So the first mechanism for that is endothelium, and our second mechanism is nerve reflex. So there are a lot of nerves that are also supplying the vessel. Like here is the nerve, here's the nerve, here's the nerve, and when the vessel is damaged, these nerves get stimulated, and they will then cause a reflex that will cause vasoconstriction. So the first step was vasoconstriction. Now, number second step is a platelet adhesion. So now, if we have this endothelium here, and this area is damaged, and then we have endothelium, the remaining of the endothelium here, then just below it, the subendothelial area, we have something called the von Willebrand factor, and this is present below the endothelium, subendothelial area. So when this area is damaged, the subendothelial collagen and the von Willebrand factor are exposed, and we know that in the circulation we have something called platelets that are floating around along with the other cells and all the content of the blood. So let's say this is the platelet, and when platelet encounter this damaged area, then it has certain receptors on top of it, and these receptors are, let's see, these blue receptors, and these are GP1B receptors, and they will kind of bind to the von Willebrand factor. And when they bind to the von Willebrand factor, that is basically what platelet adhesion. That is how the platelet adhesion occurs. So vessel is damaged, von Willebrand factor is exposed, and von Willebrand factor has receptors for GP1B to connect to GP1B, and GP1B receptors are present on the platelet. So this is platelet adhesion. After platelet adhesion comes number third step. And number third step is it involves another component of the blood, and that is fibrinogen. So in the blood we have something called the fibrinogen. So for example, this and this and this and this, and on top of the platelets there are another set of receptors, and these are called GP2B3A receptors. So these are the GP2B3A receptors, and they bind to to the fibrinogen, and the fibrinogen in turn binds to the. So there is another platelet, and this fibrinogen will bind to GP2B3A receptors of that platelet, and then in turn, this, for example, this is another fibrinogen. It will bind to another like GP2B3A receptor, and then it will bind to another GP2B3A receptor of another platelet. So you get the idea. It is kind of linking. The fibrinogen act as a linking molecule between the platelets using GP2B3A receptors. So another thing that is important is that when the platelet adhesion occurs, the platelet kind of induces a shape change in the platelet, and platelet get activated, and they release ADP, and the ADP then induces. So ADP is released. ADP induces GP2B3A receptors on top of the platelets, and then that GP2B3A receptor, as we talked about, help in the platelet aggregation. So basically, the platelet aggregation was a third step. So the third step was platelet aggregation, and we talked about that. So all of this platelet adhesion and aggregation, they will eventually form a platelet plug here, and that platelet plug is weak. So there's something needed that will kind of stabilize that platelet plug, and that is the coagulation system, and that is called secondary hemostasis. Now, coming down to various disorders that can be associated with this platelet with the primary hemostasis. So the first is a defect with the GP1B receptors. So these are GP1B receptors, and that defect is the disorder is called Bernard-Soulier disease. Bernard-Soulier disease. And it is actually qualitative disorder because there is a defect with GP1B. Although it is classified as qualitative, but it is associated with mild thrombocytopenia. And another finding that is associated with Bernard-Soulier disease is are large platelets. So that's about Bernard-Soulier disease. And then another important test that is usually tested is the Ristocetin test. So for Ristocetin test is basically what it does is it looks if the platelet adhesion is intact. And for platelet adhesion, we require two things. The first is the GP1B receptors, and number second is the von Willebrand factor. So Ristocetin will be defective when there is like absent or decreased activity of von Willebrand factor, for example, von Willebrand factor disease, or if there is GP1B defect like Bernard-Soulier. So Bernard-Soulier will have abnormal Ristocetin test. And von Willebrand disease will also have one abnormal Ristocetin test. So then so that was your GP1B, and then let's talk about the other receptor, GP2B3A receptors, and that is defective in Glanzmann thrombosthenia. So it is also a qualitative platelet defect because the platelet number is normal. So then let's talk about the von Willebrand factor disease, because that is important as well. So von Willebrand disease it is an autosomal dominant disorder, and it is fairly common, usually asymptomatic, but a symptom do occur there because the platelet kind of don't function well, because as we talked about, in order to adhere to subendothelium, it requires the von Willebrand factor for that function, for adhesion to endothelium. So when there's von Willebrand factor disease, there will be a lack of platelet endothelial adhesion, and that will result in ecchymosis and mucocutaneous bleeding, like epistaxis, and so that is how it is going to present with. But usually it is mainly asymptomatic. Another thing that von Willebrand factor it is going to present with on a lab finding is elevated PTT. So why is that? That is because of factor factor 8 deficiency. And the reason for that is because von Willebrand factor normally, von Willebrand factor stabilizes factor 8 and it prevents its destruction. So it acts as a carrier protein basically. So for factor 8, and when factor 8 is deficient, then it will result in elevated PTT. So there is in the absence of von Willebrand factor, there is increased destruction of factor 8. So one interesting thing about factor 8 is that it is produced by the endothelial cells. Okay, so endothelial cells have factor 8 that is synthesized in them, while all the other factors, most of the factors are synthesized in the liver. So that's interesting. So then von Willebrand factor, we can use a drug in von Willebrand factor disease, and that is desmopressin. And what it does is that it pushes the factor 8 that is still remaining in the endothelium out into the blood, and so that would correct the PTT. So that's important thing to be aware of. Then another thing is the source of von Willebrand factor. So where is von Willebrand factor located, or what are the sources of von Willebrand factor? So von Willebrand factor is present in number 1, platelet, the alpha granules, and number 2 is the Wobble-Palade bodies. Wobble-Palade bodies in the endothelium, that are located in the endothelium. So in Wobble-Palade bodies, if this is a endothelium, endothelial cell, in it we have a Wobble-Palade body. Let's say these are Wobble-Palade bodies, and they produce two things. Number first is the von Willebrand factor, and number third is p-selectin. The second is p-selectin, and the p-selectin kind of helps in the adhesion. So if this is a p-selectin, let's say, and this is a leukocyte, the leukocyte kind of binds to the p-selectin, and that way it helps it to get into the site of inflammation, which is below the endothelium in the interstitial space. So this is what the function of p-selectin is. So I hope this was helpful, the primary hemostasis and associated disorders.